you so much for coming along to this session. Hopefully you were at the first one. If you weren't, it doesn't matter because each is it's a standalone. So we're going to be, anyone who's been attracted by the title 50 Shades of Grey but didn't quite know, and I think this might be quite a sexy presentation. Sorry, I've got to let you down on that, as it is grey matter. And my name is Stephen Gomez, and for those who weren't at the first talk, I'm retired now, but I've had quite a lengthy time in looking at neurology. I working backwards, retired just before COVID, and to travel the world, and then there was lockdown, so it was such bad timing on my behalf, and then I came out of retirement and I helped with the NHS during the COVID time, so vaccinating, I don't know, thousands of people. Before then, I've sort of worked in hospitals and universities, training other people in neurology, in neuroscience, and then before that, gosh, I still remember, I was working at the National Hospital for Nervous Diseases, the premier place for neurological disorders, which is based in London. Neurological cases, they used to fly them in from all over the world for our attention. And before that, I was at the Institute of Psychiatry, not as a patient, I must quickly add, but working there for a little while. And before that, I graduated from King's College in London.
Just walk past, it's okay.
Before that, I was 3 years old. So I obviously stole that joke from the captain. So if you were asked to stall. So last time, there was a session where there's a whole load of things that people have got wrong about the brain. Things like we only use 10% of the brain, which is number one. Mythology about the brain. And another is about us being divided, brain being divided into left and right sides. And you're sort of left-brained or right-brained. It's a total nonsense. There is nothing that supports that. In this session, we're going to have a look at how the brain works, obviously a shortened version. If I were giving this topic to medical students, it would go on for the whole year. So I'm obviously going to take a whole load of shortcuts, but I want it to be accessible and of use to you as well. So I'll be pointing out things. I've been told that I have to really finish within about 15 minutes and include some question time in that, which I hope to do. But I am wandering around the ship, at all other times. So please, if you have a question and you want a bit more time, even if it's on a topic I'm not going to cover, please stop me and ask me. I'll be more than happy to spend that time. So don't feel as though you've got to rush things now. If there aren't any questions, just stop me and ask. So a couple of days ago, when I was talking about my introduction to the brain, I gave a number, and it was a magic number of how many neurons that we have in our nervous system, especially the brain. And it was a fantastic number. It was 10 to the power 10 neurons that we have in the brain. And that is 10 with 10 zeros after it. So it's a fantastic number. And I said it's roughly the same number as the stars in the known universe. So, there's a whole lot of stars out there, and all of this is packed inside of us. And this is a diagram. It's not exactly how it looks, but it gives a fair idea of one of these neurons. And you might think, what on earth is going on? There seems to be on fire in certain places. So we use electrical energy in order to have transmission of information. We also use chemical energy. So this gives an idea of a neuron that is active. and so there's electrical activity going on. Unfortunately, it doesn't quite look like this. If you were to look, it doesn't look exactly like this, but this definitely gives a picture of this. So in this session, I'm going to give you some of the basics of the nervous system that you need for the other 3 sessions, because I'll be referring to some of these. So this is a bit like teaching someone the ABC, and you learn all the letters of the alphabet, and then you find out ways how you can arrange these letters to form the most amazing things, novels, poems, all sorts of things. So, and don't think that, oh my goodness, I've forgotten this by the end of the session, because in one of the sessions we're going to look at memory, and I'm going to tell you then, as a preview, that you remember everything. Everything that you give your attention to is locked away inside here. Now you might say that's total rubbish. I walked away from Stephen's talk and I can't remember a thing. That is another matter. That's another process, and that's called recall. So memory and recall are 2 different processes that I'm going to go into. So just because you can't remember something, can't recall something, doesn't mean that you haven't memorized it. It is there deep inside, and there's various techniques that you can use to get that out. And by the way, I'm a mainstream scientist. So everything I'm going to talk about is mainstream. There is obviously alternative medicine and alternative therapies. I am not qualified to talk about any of those because I'm not a practitioner of those. So, but I'm happy if any of you are into alternative, I'm happy to see how the 2 overlap. We can discuss it. If we were to look at a diagram, a simplified diagram of a neuron, this is what we see. And on the far left over there is the cell body. And the cell body contains the nucleus. And the nucleus contains the DNA of how that neuron is formed. Now, we all know about DNA, that it's a blueprint of our body. Every cell in our body contains a blueprint of how to make the whole body. So you probably heard of cloning and things like that. In theory, it's not possible at the moment, but in theory it is, you can take one cell and from it get instructions of how to form the rest of the body. So the nucleus, it's the DNA, and it has a blueprint not only to produce this cell, but every other cell. Now there is one exception. All our cells in our body contains this blueprint, except one lot of cells, and those are red blood cells. So red blood cells don't have a nucleus. So if you're trying to clone someone, don't take their red blood cells, okay? Because it'll be useless. Then from the cell body is this long fibre coming out, and that's called the axon, and the axon conducts signals all the way along from the cell body. It's the cell body that makes the decision. Shall I act or shall I not? Shall I fire or shall I not? Once it's made that decision, and a bit later on I'll be hopefully telling you in the series how that decision is made. If it decides to fire, an electrical signal will go along that axon, and it travels at a fantastic speed. It travels at between 40 and 60 meters per second. So if you can imagine the longest axon we have in our body, depending on how tall you are, if you're relatively tall, if you are tall, the longest axon is the one that starts at the bottom of your spinal cord and goes all down your leg in the sciatic nerve. It goes all the way to your toes. So that's the longest one. Depending on your height, it's about 1 meter long. And so, you know, if signals are travelling along at, say, 60 metres per second, it takes no time to get there. The shorter the axon, the sooner the message gets to the other side. And so if any of you have tried spotting a fly, and, you sort of creep up on it, and you go to spot it, sorry, I shouldn't say spot it, as you go to capture it, to put it outside, then you know that they just go off. They just go off. And the reason is that because it's a fly, it's very small, its axons are very short. So by the time the message gets to the other side, it's extremely fast. So they look at us and they see us moving in slow motion. And they can think, well, we can sit here for a while, until, it gets relatively close. And so you might try to whack it or remove it as fast as possible, but it's laughing at you. If there's any entomologists in here and they tell me, in fact, flies don't laugh, I'd have to change your mind about that. And then you have right at the end, looking at it on the right-hand side, you have the nerve terminals. And this is where the signals get to, and then it causes the release of a chemical at the end. And that chemical goes and activates whatever its target is. It could be another neuron, it could be a muscle, it could be a gland, or whatever. So this particular cell has been, I've been studying it, especially the nerve terminals right at the end, and the most remarkable things happen, and I've been studying that for decades, and I still don't know everything about it. So you can imagine that's like just the very end of this. The axon is insulated in order to make it go fast, and in order to make sure that the signal doesn't creep into another axon going somewhere else. It's insulated, and it's insulated by a fatty substance called myelin, and it's not a continuous insulation, but there's gaps. And what happens is that the signal doesn't go along continuously, it jumps. And the person who actually found that and found these gaps is a fellow called Schwann, and it's one of the many places in the body where there's a name for something that doesn't exist. So where there's a gap, then he's named it after himself. Nowadays, you don't tend to name discoveries after yourself, unfortunately, because people don't understand exactly where it comes from, but it's named more anatomically. We don't just have one neuron, obviously, but we have networks. And here, a series of these axons, of these neurons, have been stained with a dye that fluoresces, that shines, and so we can have a look at that. This is nowhere near the complexity of what actually exists. And the sort of pattern we see in our neural networks are unique to us. The pattern of these neural networks is us, is our personalities. We all differ, we all have different experiences, and I said in the first talk that we are formed by what we lose. We are formed by neurons that are lost. And the ones that are remaining is us. Is our character, is our behaviour, our reactions to things. There's obviously a lot of overlap because we're different, but we're very similar in many ways. We react, so there is a bit of overlap, but the actual precise arrangement of all of this is unique to us. So if you're looking at this and think, oh, this is really complicated, I can't get my head around it, your head is around it. So this is you. If we take out, I'll try not to make the pictures too graphic, but anyway, if you want to look away, this is a brain that's been taken from a patient that's died. And usually when we remove the brain has the consistency of stiff porridge. And like people who have actually seen a human brain like this, they go, "That's ridiculous, that's rubbish," because I've held the brain and it's not that stiff porridge at all. It's really hard. And I say, have you held a brain that's recently been taken out of someone, or one that's fixed? And so brains, because they're so friable, they're so delicate, they're put in formaldehyde, in order to strengthen them up. So then you can do various things with this. So this is a brain, and you can see all the curly bits on top. And the cerebrum, the human cerebrum, makes up, and that's the bit sort of highlighted in red, makes up 80% of the brain. Now, we are a species that is way above all other species as far as cognitive abilities, as far as just our memory, synthesizing new material, all sorts of new thoughts. We are way above all other animals. If you plot a graph showing the development of certain bits of the body, it's more or less a line that goes up continuously, and then we're sort of there. If you plot the development of the brain, then of various animals, and then it goes up, and we are an outlying point. We are not actually on that curve. We are somewhere way above, and there doesn't seem to be any connection between even our closest relatives, the primates, and us, as far as cognitive ability is concerned. There isn't a gradual development to us. We have leapt. Why that has happened, how that has happened, is our discussion now, because we have goes into lots of things. But to just keep that in mind. The surface of the cerebrum is wrinkled. And whenever you see something wrinkled and not flat, you immediately think this is to increase the surface area. So as our brains developed, as we became reach high levels of cognition, the development of us had to make a decision. Does the brain and the head get bigger? Like you see some aliens, on sci-fi programs, they've got enormous heads. So in our development, a choice was made: do our heads get bigger, or do our heads stay roughly the same size, and it gets more crinkled and you get more surface area? And I don't have to tell you the direction that we went in. And the thing that limited us, the reason we didn't go down the big-headed route, was that we wouldn't be able to be born. There is a limit to how much of a charge you can get out in one day. So, we decided to keep, our development decided to keep the heads the same size roughly. And with a baby's head, it's quite enormous, considering, it's out of proportion to how it is, say, in an adult. And again, if you were the first talk, I mentioned that the first thing to develop in the fetus, in embryo, is the nervous system, because the nervous system controls everything else, and it lays down its fibres everywhere in readiness for the tissues that it's going to innervate to get into contact with. And I said that it forms in the first 3 to 4 weeks. So often before the woman knows she's pregnant, the nervous system, the rudiments of it have been laid down. So the cerebrum, as we're going to see, contains numerous centres. You might not think that there's all these centres there, but I'm going to introduce you to a few. Keep an eye on the time. It's within the cerebrum that we have our cognition. So not our memory, when you're working out things, should I go to Stephen's talk or shouldn't I? All those sorts of things are decided in the cerebrum by those neurons that I mentioned. And we have self-awareness. So we know we're here, we know we're with other people and all because of that. And I'm going to be talking a little bit about that when I talk about sleep and what sleep is from a physiological point of view. So if you do have any questions, if you could just mentally keep them or write them down to make sure you don't forget, and definitely ask me questions afterwards. So if you were to open up someone, then their head, again, I don't recommend this, but just believe me, this is the case. And if you were to look down, you would see that the brain is divided into two. It's divided into two hemispheres. And why it should be like that is a story not for today. But it's divided into left and right. And left and right is always the patient's left and right. I get confused by left and right when I look at the screen, because it's obviously different from here. But the number of things, of treatments that have gone wrong, because if you're looking at the patient, you're facing them, your right is their left. And so you know there's this convention, there's always the patient's left and right, not yours. So in some operations and all, you think that the surgeon is like the person who's sort of in charge. It's the theatre nurses, and quite often you sort of go in there and you know and then it's the other arm, so just make sure that in the right. If ever I went in, I don't have any tattoos, but if I did have to go in, I would have arrows pointing, cut here and what to do. The strange thing is, well, there's lots of strange things with this arrangement, but one is that one half of the brain controls the opposite half of the body. So why this should happen, again, is another story, is a story for another day. But at some point in our evolution, each side did control the same side, but it twisted over as we went through evolution, went through earlier stages. So curiously, if you know someone, unfortunately, who's had a stroke or a problem, let's say affected their right side, then you know it's their left side of the brain that is affected. So there is this crossover of action. So people who were proponents of left brain, right brain, if you're scientific you're one, if you're artistic you're another, rubbish. They looked at the brain. They said, two halves. There is a left and a right, and so therefore they must be separated for some reason. It is separated for some reason, but not that reason. But the two are interconnected by this band of fibres called the corpus callosum. So whatever happens in one half, it connects to the other. And the last time I mentioned, sometimes surgically, that's cut. And in cases of epilepsy, where you have a focus, say, starting on one side of the brain, it's like a wildfire. It can be, depending on the type of epileptic attack you're having. It's like a wildfire that starts, and it just spreads over that hemisphere, and then it goes conveniently in the corpus callosum to the other. And so in case of really severe epilepsy, they cut the corpus callosum just to limit it at least to one side. I hope this is all making sense, by the way, to some degree. Yes? Okay. So the brain is divided into lobes. It's divided into lobes by us. Okay. If only the brain looked like this in real life, coloured in, it would be fantastic. But this is how we get students all to learn about the brain. So this is the cerebrum, and you'll see that there's different parts. Starting at the back, when I was working at the Institute of Psychiatry, I used to run courses for psychiatrists, for them to learn a bit more about the brain. I mean, they've done it as medical students, but it was a refresher course. And many of them, I had to point out what was the front of the brain. Put it in the right right direction for them. So on the right, on your right, there's, we forget about the cerebellum for the moment, but just looking at the lobes, there's a lobe called the occipital lobe. Now that lobe analyses and processes all your visual information. So when you're looking at something, this is your visual field. Whatever you're looking at is projected onto the retinas, and the retina turns that image upside down. So everything we see is upside down, and it's worse than that. So anything, so this is your peripheral vision, the bit around the edges. Everything on this side, for me, goes in through the eye, lands on the retina, and is projected on the other side. This bit over here goes through to the other side. The bit in the middle goes to the same side, and this bit goes to the same side. Can you imagine what that image looks like? And so the brain then processes that, stitches it all together, and it produces what we're looking at. So that is right at the back of the brain, the occipital lobe. And when I was a student, when I was learning about all of this, I thought that's a really crazy place for the occipital lobe. Surely it should be closer to the eyes. You know, why is it at the back? And you know, the less you actually know, the more you're sure about. So I was like, you know, thinking, well, you know, it's evolved in a really strange position. It hasn't. It's evolved in just the right position, because what happens is that so much processing goes on that it needs to be at the back, and it passes through relay stations and messages where it helps with the processing of the image. Anyone who's had a knock to the head, fallen over, and they see stars or funny patterns or anything like that, the brain, because it's made of this stiff porridge consistency, has to float. It can't rest on anything, because if it rests on anything, it'll damage it. So we have a fluid inside our craniums and the spinal cord as well, cerebrospinal fluid, and this gives it buoyancy. It allows the brain to float, and our spinal cord as well a bit. And so as we shake our heads and do things, the brain wobbles inside very, very slightly. But if you have a traumatic impact, say on the front of your head, and it forces your head back, there's a slight delay with your brain because it's in this fluid. And so as your head goes back, the brain follows after a short while, and it strikes the back of the cranium, and it activates the cells in the occipital lobe, which is why we don't see an image as such. I mean, you could do, but you know, usually it's strange lights and things like that. The temporal lobe is controls a lot of our emotions and our feelings and things like that. It connects with that. And you've heard of lobotomy, where bits of the brain are removed. And a lobotomy was, thankfully not anymore, it was performed in patients who are particularly violent, have really bad behaviour and uncontrollable behaviour. And what they used to do is to remove the temporal lobe or part of it from patients. I mean, this is barbaric, look on it, but this is what standard procedure was in those days. So how would you remove a part of the brain? And the answer is that you drill a hole, a burr hole, and you drill a hole with a drill. In those days, a hand drill. And once you made the hole, you put in a tube, and you could suck away the temporal lobe. Because the brain is like stiff porridge, you can suck it away. It was something like the diameter of a McDonald's straw. Again, I'm not recommending you try this, but you can actually do this. The frontal part of the brain it controls our behaviour as well, and this is our cognition. The frontal part of the brain is the last part of the brain to develop. Compared with other animals, we are the slowest developing. Perhaps there's a tortoise or something somewhere that has a slower development than us, but you know, of all the animals, primates and all, we take ages for our brains to develop. And the frontal part of the brain doesn't develop fully until you're about 25. And so with children, I used to foster children, and you know, I used to look at their behaviour. Their behaviour was particularly bad, and I used to describe the neurology of some of these kids. It wasn't my job, but I thought I had to. And their brains, the frontal part, just wasn't developed. But most children are like that. Children are very mean. They're very self-centered, and the reason is that they've developed like that because of survival. If you want to survive, you don't share anything, because that's ridiculous. It affects you. So you want to hold things to yourself to make sure that it's not shared. And children have not learned the basis. As adults, we know that it is important to share, because there's lots of benefits in sharing. You know, and I have a young person who one of the kids I used to foster, still in touch. He's 22 now, so you know, like he's well over 18, and he calls himself an adult. But he comes over to my house. Anything of mine is his. Anything of his is his. You know, and I tell the story of, like, I was searching around for a biscuit once. I didn't have and he had a packet of biscuits there, which he left. I had one biscuit from him. Then I remember him come back to, "Have you been at my biscuits?" You know, and like that amount of stuff I give him. So you know, and I think his frontal cortex has not developed. But I can see stages where he's becoming more empathic. So there is hope on the horizon. So I'm not going to go into all of this in enormous detail, but you have the cerebrum on the outside, into this bluish grey, and then you have a number of structures. So working from the bottom is the spinal cord, and the spinal cord comes up and it forms a large area, the brainstem, and the brainstem is vital for our survival. If the brainstem is damaged, then it's brainstem death. Death used to be recorded in a number of ways. In the past, before people really understood about the brain, death was defined by you stopping breathing. Or death was defined by you stopping breathing, or death was defined by your heart stopping. Now it's defined by the brainstem being damaged. So we're going to see why. And above the brainstem, you find the midbrain, and this contains areas to do with your emotions. So any emotion you have originates in that part of the midbrain. And there's bits there also. The pituitary gland. The pituitary gland is known as the master gland of all your hormones. So all your endocrine, all your hormonal glands around the body is controlled by this tiny pituitary area, and so it's known as the master gland. The master gland has its own master, and the master is the hypothalamus. So the hypothalamus controls that, and it controls a lot about you. When you get thirsty, it's your hypothalamus saying, it's predicting that your fluid levels are going down. You're getting hungry, it detects that your glucose is going down, and it's got so many important functions. It's a tiny little area, and it's about 5 grams in weight, so it's a very small. So much there to talk about, but anyway. So we have this this nervous system. It's divided into two. There's the central nervous system, which is the brain, brain stem, all of that area, and then you have the spinal cord, which goes down. And from the spinal cord, you have all these nerves coming out, and they come out in between your vertebrae. And so I can draw a map, or I haven't got a map here, but there's a map of your body which is innervated by spinal nerves that go out. And so in order to test whether your spinal nerves are okay, you've also seen physicians with a pin, and they would touch your skin, and they would say, "Oh yes, can you feel that?" Yes, and then they would go to another area and say, "No, I can't." And that's how they can find out this particular part of your spinal cord that's affected. So that's the spinal cord. We're going to just have a quick look at the cranial nerves, because these are nerves that grow out from the brain that innervate everything in your head and neck. So they're supplied just by nerves that grow out. So for instance, smell, there's 12 pairs of these cranial nerves. I'm not going to go through all of them, don't worry. But there's some, number one is the one that affects our smell. So people often think sight is one of the most important senses, which it is, but you detect quite a lot of things subconsciously through smell as well. That has a direct, so it's number one, cranial goes directly to the brain. There's a whole load of other ones. There's the optic nerve, which forms the eye. And I remember driving to work once, and there was a quiz on like one of these radio stations, and it was where do you find the retina? And so the person was saying retina. I'm sure I heard of it. I'm not quite sure. Is it the brain? And the person said, no, it's not the brain. It's the eye. And I felt like phoning in actually and saying, well, actually, it's technically the brain. My students would have liked, oh God, it's him again. He's on the radio now. I didn't call in. But what happens is, again, in our fetal development, you have stalks growing from the inside of the brain, and it grows out and it grows out in the developing fetus. And you can imagine it just like a stalk, and it hits where a part of your, so it's trying to get to the surface, it hits part of your face that is blocked off, that will eventually become your eye socket. It hits that, it splays out like that, and it forms the retina. And the bit it hits will form the lens of your eye. How beautiful is that? And not only that, there are 1,000,001 things happening elsewhere. That's just one of the things. And not only that, it happens in concert with the other eye. So it all goes amazingly well. In the majority of cases, there are cases where it doesn't quite work out like that. Again, I could wax lyrical, I think, on all of these, but I'm going to force myself to carry on. So I mentioned something about spinal nerves. Let's have a quick look. Oh gosh. So what do we look like to our brains? To our brain. And this is what we look like to our brain. We don't look like this. We look like this distorted image where we have a large face, we have really big lips, a big chin, enormous hands and enormous feet. And if you were isolated from everything else, this is how you would imagine yourself to be. But when we look at ourselves and we see we're not like that, that overrides our brain's image of it. And the reason we look like this is known as a homunculus. And an homunculus has these large features on the face because it's associated with how sensitive certain bits of the body are. So our lips are much more sensitive than our backs or parts of our arms. That's why you can get a little nick in your lip, and it is painful. And you look at it and you can hardly see it. And our feet are enormous in this. And when you're walking along and there's a piece of grit in your shoe and it's crippling you, you cannot carry on walking. You have to stop. You stop, you take off your shoe to get rid of this enormous rock inside your shoe, and you're there for ages, and out comes this tiny little grain of something, and you think, this has crippled me. So, just going to go on, if that's okay, for about 10 more minutes, and then take questions. So there's lots of things in the brain and the brainstem. One is the respiratory centre. So the reason why death is defined by your brainstem not working is that there are neurons in there. There's like those neurons we saw earlier that rhythmically cause us to breathe. And breathing is it's passive in a way, in that we're drawing air in from the outside. And we're doing it in a way that we're almost trying to cause a vacuum inside us. And so we increase the space. We have nerves that go to the intercostal muscles. And the intercostal muscles are, if you've eaten spare ribs or ribs, those are the intercostal muscles. And they're delicious. Not in humans, but in animals. And it causes the ribs to move outwards. And when the ribs move outwards, it creates a space inside our lungs. Our lungs are there just flapping. They can actually pull in air themselves. And we have another set that goes to the diaphragm. And the diaphragm is this dome muscle. And the diaphragm is pulled down, it's made flatter. And so both those actions cause air to be drawn in. Again, there's lots I can say about that. Whenever you cough, it's centres in your brain that coordinate the coughing, because we need to breathe. Coughing is going to interrupt our breathing, and it needs to be coordinated. Many of the medicines that you take, especially if you have a severe cough, goes to the brain. They're called antitussives. And a tussive is anything that makes you cough, and an antitussive stops that. So cough mixture used to contain opium, used to contain quite dangerous substances that goes to our brain, and it quietens down it's a wonderful cough mixture, but terrible in all sorts of other ways. So people got addicted to it. There's centres in the brainstem that control our heart. There are centres in the brain you may not know about that controls our motivation. You're lying in bed. Shall I get up? Should I not? The decision to get up or not may be forced on you. But if you're there and you're thinking, should I get up? That's the motivation centre. And it kicks in. And when it kicks in, it causes impulses to flow around your body to move yourself and to go off. And there's a reward. There's lots of things that you think, I'm going to get up and go for breakfast. So that's going to be my reward. So that's another push. And there are centres there releasing dopamine that give you pleasure whenever you sort of like, it's the reward centre there. It also helps with decision making. And there are various things we use to explore decision making. And for instance, if I were to say to you, would you take £50 now or £100 in a week? I'm sure the majority of you would say, I wait a week and I get the £100. And then if you were asked, would you take £60 now or £90 in a week? And I think the majority of people might say, well, I'd say £90. But you get to a tipping point that you think, I think I'm going to take that now. All of that decision is made in these areas of the brain. So you might think, oh, well, I'm in control, I'm making the decision, but, and these are the reward systems that people offer. Like buy now, and you get a discount, and there is a tipping point when you decide. was going to cover, would it be okay if I carried on for just 5 minutes more? Because I was going to cover this business of scanning the brain. In my day, I trained, I practiced, and I taught at a time before computers. And so a lot of things that are available today were not available then. And I'm a great proponent of computers and using computerized methods in neurology. I'd be a fool if I didn't. This technique was brought in called computerized tomography, known as CT. Many of you might have had a CT, or you'll have a CT or know someone who's had a CT. And tomography comes from the word slice, as I mentioned last time. And what computerized, what CT is that it's a series of X-rays. So just as an ordinary X-ray that you would have. Now, before CT, you could take X-rays, so nothing was new there, but you were limited. You could only take, a few planes. This takes slices through your body, particular parts of your body, and so you could look at them in a serial way. So that's X-ray. So if you have a CT scan, and this thing revolves around you, and there's a detector, there's an emitter and a detector. Because it's X-rays, then there's obviously a few dangers associated with that. But because it takes a whole series, it can do it at a lower power because it's made up out of a number of. And so you see beautiful images like this, and you can locate tumours, you can locate various things that have gone wrong inside the body, inside the brain. And obviously you can do this in other parts of the body. I'm not interested in other parts of the body, only looking at the head. And computerized tomography was found by this guy, was made by this guy, Hansfield, and you can see an early version there. He wasn't a medical doctor, he was a bioengineer, but obviously working with medics. A massive contribution. This is such an important technique to use and device to reproduce. And the first scan was made in 1972, and he won the Nobel Prize in medicine, in physiology and medicine. You've probably heard of a PET scan, and so PET stands for a positron. That sounds like some sort of radiation emission tomography, and again, tomography is a slice, and because it's a slice and it goes around this tube, this donut, as people call it, and it uses various tracers. Now, this is better than CT for some things because it lights up various things in the body that are working. So it gives you an idea of metabolism, of how active certain areas are or how inactive they are, if it's been damaged in some way. So this is a really useful technique to use. So we'll go into more detail about this in time. And I always find that the people who develop these things are rarely given any exposure. So you go into for a PET scan, you wouldn't think, who invented this? How was this produced? But these are the couple of people who did it, Americans. And then the MRI, which is one of the latest ways, and this is a really powerful magnet, and this uses a different technique. So all the cells in your body are largely made up out of water, and what happens is that water is H2O. It's a hydrogen, two hydrogens and oxygen. And if you were to look at a water molecule, there would be a charge on one end and a different charge on the other. And it's all higgledy-piggledy. The water inside this is all higgledy-piggledy. And it goes around. You get inside this magnet, the magnet produces really powerful charge. And what it does is that it forces all of our water molecules, all of the various ions inside our body to line up And once it's lined up, it switches off because the magnet has given power to these things to go out of their usual position and to line up. And once the magnet's turned off and the power's on that, these molecules jump back into position to what they were. When they jump back into position, they give up the energy, which is then detected. How clever is that? And what happens is, it gives you a 3D image of what's going on in the body. So rather than these slices, this is not tomography, this is not these slices, but it can give you a 3D image. So you can twist it this way and that way, like a CD. So that gives you an idea of that, of something that you see. And you can colour it in all sorts of different ways. So the person who actually developed this, who has the patent on this, did not win the Nobel Prize. The Nobel Prize went to 2 other people. And this guy was so annoyed that he didn't get it, that he took out advertisements in newspapers, whole page advertisements. He spent a fortune to the complaining about this. He does hold the patent. Well, he's dead now, but he did hold the patent on all this. So he did get a lot of money, but it shows you how awards such as Nobel Prizes and all are just so important. Nobel Prize, I think, is only £250,000, which isn't an awful lot. I'm so sorry. I think I've gone over my time. If any of you have any questions, I'm happy just to wait in the back. I think there's probably something going on here, and happy to answer your questions. If not now,